Hello and welcome back to Dental PYQs. Today we'll be discussing the October 2025 Physiology paper. So let's begin. Let's see the first question. Describe the pathway of pain and add a note on referred pain. So ये है आपका 10 mark का question. So they've already divided the question into two parts. The pathway of pain will carry 5 marks and the note on referred pain will carry 5 marks. So this referred pain is very important because it has been asked n number of times previously. For example, in the December 18 paper, next it was asked in November 2020, and then it was asked in March 2021, and also in this new paper of 2025. So let's see the definition of pain. It's defined as an unpleasant and emotional experience which is associated with or without actual tissue damage. The transmission of pain sensation occurs in 3 stages as the pain is transmitted to the sensory cortex from the affected area. So the three parts or the 3 neurons, we would say, this is the first order neuron. It is from the site of transduction of pain to the dorsal horn of the spinal cord. The pain impulses will travel via the 2 fibers, that is the A delta fibers and the C fibers. A delta fibers are fast conducting fibers and these are slow conducting fibers. The difference we will see between the two in the following table. These fibers, they terminate in the dorsal horn of the spinal cord. A synapse occurs between the nociceptive fibers and the dorsal horn neurons of the spinal cord. Excitatory neurotransmitters are released. Matlab, these are neurotransmitters which will increase the action potential there by releasing nitric oxide, ATP, substance P, glutamate. CGRP is nothing but calcitonin gene related peptide, and then we have the bradykinin. These neurotransmitters will bind to the receptors and the pain impulse is transmitted across the whole synapse. Now from the dorsal horn of spinal cord, the pain impulse is transmitted to the brainstem and thalamus via the second order neurons. The pain impulse will ascend from the spinal cord to the brainstem and thalamus via the 2 nociceptive pathways, which is the spinothalamic tract and the spinoparabrachial pathway. Now the difference between these two will be seen in the table below in detail. Then we have the third order neurons, which will carry impulses from the thalamus to the cortex and the higher brain centers. They relay pain impulses to multiple cortical and subcortical areas in the brain. There is no discrete pain center in the brain, and the pain perception occurs only after integration and processing at the higher brain level. Now we'll see the table between the spinothalamic pathway and the spinoparabrachial pathway. So the alternate name of spinothalamic is the anterolateral system, and the spinoparabrachial is the spinomesencephalic and the effective pain pathway. The primary function is it discriminates between the aspects of pain and temperature, and here it is motivational and autonomic aspects of pain, basically the emotional response, as we can say it in a easy way. The type of pain conveyed is by the fast and slow pain fibers, and this is by the predominantly slow and persistent pain by the C fibers. Modality is by the pain, temperature, crude touch. Here it's associated with emotional and autonomic response. The first order neurons, the second order neurons, the desiccation and the ascending tract, all these are same for both pathways. Okay. That is, they arise from the dorsal root ganglion. It crosses in the anterior white commissure between the first and spinal segments, and it ascends in the anterolateral funiculus. The major relay that occurs between these two is the spinothalamic is at the ventral posterolateral part of the nucleus of the thalamus, and here in the parabrachial pathway is by the parabrachial nucleus in the pons. ये thalamus है and ये pons है. Then we have the third order neurons, are the last set of neurons. The primary and secondary at the somatosensory cortex, and here the parabrachial is at the amygdala, hypothalamus, insular cortex and all the other limbic structures. The level of conscious perception is high in the thalamic and low in the parabrachial. The emotional component is very minimal in the thalamic and it is very, very predominant in the parabrachial pathway. The autonomic responses are limited while they're very strong in the parabrachial pathway of the cardiovascular, respiratory and stress responses are seen here. We will understand the two pathways with the help of a diagram here. Now this is the spinal cord, dorsal. This is the brainstem. This is the midbrain. And this is the higher brain center. Okay, so the red tract that you see here is from the superior dorsal horn here. It crosses all the way and it relays in the thalamus. So this is your spinothalamic tract. Okay. Then the blue line that you see is from the superior horn of the dorsal part of the spinal cord. It goes all the way up here and it relays in the amygdala and VMH. VMH is nothing but the ventral medial nucleus of the hypothalamus. So the one that is relaying in the hypothalamus and amygdala is your spinoparabrachial tract. This was a little complicated. If you can draw that, you can continue to draw. If you want a little easier, simpler diagram of just the pathway of pain without the two tracts, you can draw this. Here we have the nociceptor or the pain receptor to the dorsal horn of the spinal cord. So this will be your first order neuron. Then we have the pathway from the dorsal horn of the neuron all the way up to the thalamus. This will be your second order neuron. And then from here, from the thalamus to the higher brain centers, to the higher brain, we have the third order neuron. Now we'll see the difference between the A delta fibers and the C fibers. The A delta fibers are fast conducting fibers while the C fibers are the slow fibers. The reason being that they are myelinated and these are unmyelinated. The conduction velocity is about 5 to 15 in the A delta fibers and about 0.5 to 2 in the slow pathway. The type of pain here in the A delta fibers is sharp, well localized pain, while it is very dull, diffuse and aching in the C delta fibers. The name of the pathway will be the neo-spinothalamic pathway for the fast pathway or the A delta fibers, and we have the paleo-spinothalamic pathway for the C fibers. The main tract is the spinothalamic for the fast pain pathway and the spinoreticular tract for the C fibers. The site of pain perception is at the postcentral gyrus for the fast pathway and it's not precisely located for the slow pathway or for the C fibers. Coming to the next part of the 10 marker, referred pain, which is again asked for 5 marks. Okay, it is the pain that is perceived at a site that is to or away from the site of origin. The reason for this pain is being that the viscera is often referred to the region of the body of the surface which is supplied by the posterior root of the same spinal segment. That means where the neurons relay at the dorsal horn of the spinal cord. So we have the three theories that will explain us the referred pain. We have the dermatomal rule theory, the convergence theory and the facilitation theory. The first theory is by the pain is referred to a dermatome with the same embryological origin. The convergence theory states that the visceral and the somatic efferents that are present, they converge in the same spinothalamic neuron. The facilitation theory states that the visceral impulses, they lower the pain threshold of the spinothalamic neurons. The key mechanism in the dermatomal is that it's of same embryonic origin. Here it is because of the shared spinal neurons which receive both the impulses. And here we have the subliminal fringe which increase the neuron excitability. The site involved will be the spinal segment, here the dermatome. Here we have the spinothalamic tract. This is also by the spinothalamic tract. The type of efferents involved will be visceral to somatic, that to a segmental. Here will be visceral and somatic, the efferents, that is the input neurons. And here we have the visceral efferents which will facilitate the somatic pathway. The neural threshold is normal in the dermatomal and convergence theory, while the neural threshold is drastically decreased in the facilitation theory. The reason for the pain mislocalization or reference is that the brain will interpret pain according to the dermatome. Here, the brain cannot differentiate between the visceral and the somatic input, and in the facilitation theory, we have the minor somatic stimuli which becomes effective. The nature of pain is well defined in the dermatomal theory. Here the somatic area is supplied by the same segment, and here the somatic area is supplied with an exaggerated pain. The example in the dermatomal role theory is the heart pain is referred to the left arm, and the pain of the ureter is referred to the testis. Here in the convergence theory, we have the visceral irritation, which gives rise to the somatic pain, and here any visceral disease will reach to the hypersensitivity in the skin area. The clinical significance of each theory is that the dermatomal role theory, it helps localize the visceral disease. Here it explains the mechanism of the referred pain, and the facilitation theory explains the increased sensitivity in the referred area. So this covers your 10 marker on the pain pathway and a note on referred pain. So let's see the next question. For 5 marks, we have the conducting system of the heart. This was asked previously in December 2016. The conducting system of the heart is also called as the junctional tissues. It's formed by the modified cardiac muscle fibers. It comprises of the AV node or the atrioventricular node, the bundle of His, the right and left bundle branches, and the Purkinje fibers. We have the SA node or the sinoatrial node. It's located in the right atrium just below the opening of the superior vena cava. The AV node lies in the right posterior part of the interarterial septum. The impulses, they originate from the SA node and they spread to both the right and the left atria. These impulses will reach the AV node via specialized internodal fibers. ये थोड़ा key point है, but it may fetch you extra marks if you write. So the 3 internodal fibers are the anterior fibers by the Bachmann, the middle internodal fibers of Wenckebach, and the posterior internodal fibers of Thorel. So these fibers from the SA node will converge onto the AV node and they interdigitate with the fibers of the AV node. From the AV node, we have the fibers of bundle of His which arises. This bundle of His will divide into the right and left bundle branches. These branches will run on either side of the interventricular septum, and from each branch of the bundle of His, many Purkinje fibers will arise and they spread all over the ventricular myocardium. So to summarize, we can say we have SA node which generates the impulses. It passes on to the AV node, the bundle of His, to the right and left bundle branches, the Purkinje fibers, and the ventricular contractions occur. So the SA node, it is located at the right atrium near the opening of the superior vena cava. Its function is the pacemaker of the heart, or it generates the electrical impulses. Then we have the AV node, also called as the atrioventricular node. It is present in the right posterior part of the interarterial septum near the coronary sinus. It receives arterial impulses and it's responsible for delaying them before the ventricular conduction happens. Now this delay occurs so that the atria are completely emptied before the ventricular contraction starts. Then we have the bundle of His or the AV bundle. It extends from the AV node into the interventricular septum. It conducts impulses from the atria to the ventricles. Then we have the right and left bundle branches. They are present all along the interventricular septum and they carry impulses to the respective ventricles. Then we have the Purkinje fibers. They are present in the subendocardial layer of the ventricles. They rapidly distribute the impulses, causing coordinated ventricular contraction. Then you can draw a diagram. This that you see here is your SA node. Then we have the internodal fibers. ये वाले 3. You can see the 3 internodal fibers. This is your AV node. From the AV node, we have the bundle of His. Now this bundle of His will divide into the right and the left, right plus left branches. And these branches, the smaller thin fibers that you see here, all over are your Purkinje fibers. So this covers your 5 marker on conducting system of heart. Let's see the next question. Describe the intrinsic pathway of coagulation. This is a 5 marker. This is also asked previously in the year November 2021. Okay, we will see the definition of coagulation first. Coagulation or clotting is defined as the process in which the blood loses its fluidity and becomes a jelly-like mass few minutes after it is shed out or collected in a container. We have the stages of blood clotting. We have firstly the formation of prothrombin activator, conversion of prothrombin to thrombin, and conversion of fibrinogen to fibrin. Now the formation of prothrombin activator occurs by the two pathways, that is the intrinsic and extrinsic pathway. The intrinsic pathway for formation of the prothrombin activator, it's also called as the intrinsic because the formation of prothrombin activator is initiated by the platelets itself, which are present in the blood. So it's called as the intrinsic pathway. Let's see the sequence now. So whenever there is an injury, the blood vessel is ruptured and the endothelium is damaged and the collagen that is present below the endothelium is exposed. Now this exposed endothelium is negatively charged. So when platelets that are positively charged come in contact with this collagen of the blood vessel, it gets activated and it releases the phospholipids. Now when the clotting factor 12 or the Heckman factor, it comes in contact with the collagen, it's converted into an activated factor in the presence of caliculin and also the high molecular weight kininogen. Now the activated factor 12 will convert the factor 11 into an activated factor in the presence of kininogen again. Now the activated factor 11 will convert the factor 9 into an activated factor in the presence of factor 4, also called as the calcium. This activated factor 9 will convert 10 into an activated factor in the presence of factor 8 and calcium. Now the factor 10 will react with the platelet phospholipid and factor 5 to form the prothrombin activator in the presence of calcium ions, and the factor 5 is also activated by the positive feedback effect of thrombin. We will see this with the help of a diagram. So we'll draw a flowchart of the intrinsic pathway. We have precalcinin, which is activated by the factor 12, which is already present. Okay, it gets converted to calcinin in the presence of kininogen. These two will activate factor 12 to 12A. Now this activated factor 12 will activate the factor 11. Now this factor 11 in the presence of calcium will activate the factor 9. Now the factor 9 in the presence of phospholipids, calcium and factor 8 will activate the factor 10. Now this factor 10 is like a common point, a midpoint for both the intrinsic pathway and the extrinsic pathway. This factor 10 in presence of factor 5, calcium and phospholipid will activate the prothrombin, say thrombin. Now this thrombin will activate the fibrinogen and convert it into fibrin. So this was your 5 marker on intrinsic pathway of coagulation. Let's see the next 5 marker on endometrial changes during the menstrual cycle. This question was asked in the July 2015 paper. Menstrual comes from a Latin word which means mensis, a month. This is because the lunar month of 28 days. So we will see the definition. Menstrual change is nothing but the cyclic changes that occur in the endometrium during the active reproduction period, that is from menarche to menopause in females, which leads to the recurrent monthly bleeding per vaginum. So we will see this under the 4 different phases and also under the influence of the pituitary gland. the changes in the ovary and the changes in the uterus. Okay, so first we have the day of menses, which lasts from the first day to fourth or fifth day. The FSH and the luteinizing hormone are relatively low. There is degeneration of the corpus luteum from the previous cycle, and the estrogen and progesterone levels fall drastically. Now, because of this fall in the progesterone levels, the FSH hormone and the luteinizing hormone begin to increase. The changes in the uterus that are seen is because of the falling levels of progesterone, there is endometrial shedding, and the menstrual bleeding occurs, which is followed by repair of endometrium. The next we have the proliferative phase, the estrogenal phase, or the pre-ovulatory phase. This lasts from the 4th or 5th day to the 14th day. Here the follicular stimulating hormone and the luteinizing hormone are slightly elevated. Okay. During ovulation, there is a marked increase in the luteinizing hormone because of the increase in the estrogen. Now the estrogen is increased because of the developing follicles. In the uterus, we have the endometrial proliferation occurs. That means the endometrium starts to thicken. Now here the endometrium was shed. So here it starts to thicken. It's about 2 mm. Then we have the tubal glands start to develop, and the progesterone receptors increase because of the developing follicles. Then we have the phase of ovulation. This is considered as the best stage to conceive. Okay. Now what happens here is that in the ovaries we have the follicular maturation which is happening and ovulation starts, because of which there is a lot of FSH secreted, estrogen levels increase drastically, and the LSH, the luteinizing hormone, and FSH will increase because of the positive feedback mechanism. It's a loop. The endometrium will continue to proliferate under the estrogen. That means the tubal glands will develop, and the arteries that are developing will become tortuous. Then we have the secretory phase, or also called as the implantation stage. Here in the pituitary, we have the high estrogen and progesterone levels, which will inhibit the FSH and luteinizing hormone. The follicle here becomes the corpus luteum, which secretes high progesterone levels and moderate levels of estrogen. If there is no fertilization that is happening, this corpus luteum will degenerate it by the end of the secretory phase at about the 25th day. Here in the uterus, because of the progesterone levels that surge very high, the endometrial thickening happens, which becomes about 6 mm. The glands they develop, that is, they become more tortuous. The arterial supply of the veins and arteries, they become to about 75%. They reach their maximum here. And also we have the vaginal secretions which are there, which increase. At the end of the cycle, if the progesterone levels are falling and it is not fertilized, the degeneration begins. Then on the next menstrual cycle begins at about day 1 to day 4 and 5, same as the first one. Estrogen and progesterone levels, they fall very low. The FSH will start to increase because of the decline in the progesterone levels, and there is endometrial sloughing occurs and menstruation begins. The whole of the endometrium is shed, leaving behind about 2 mm for the next cycle to begin. Now you can draw a graph. Now, as you see, at the ovulation phase, the luteinizing hormone and FSH are at peak. Okay. Now, as the progesterone levels will increase, they start to decrease. The body temperature also increases in the luteal phase. As you can see here, by about the 7th, say 14th day, that is the proliferative phase, these are the tubal glands and these are the arteries. They start to develop and maturation occurs by about the 14th, say 21st day. The glands become more developed and the arteries are reaching to their bases. There is also thickening of the endometrium. Here the endometrium is about 6 mm thick at the final stage. The glands are more developed and the arteries have become more tortuous. So this completes your 5 marker on the menstrual cycle. So we'll see the first 2 marker. Name the 4 proteolytic enzymes of the gastrointestinal tract. we have the four enzymes. The first one is the pepsin. It's secreted from the chief cells of the stomach, that's the gastric glands of the stomach. They initiate the protein digestion by converting proteins into proteoses and peptones. Then we have the chymotrypsin, which is secreted by the pancreas as trypsinogen. It's converted into its active form. It digests proteins and polypeptides into the smaller peptides, and it's also responsible for activating the other proteolytic enzymes. Then we have the chymotrypsin. It's again secreted by the pancreas as the chymotrypsinogen. It breaks down proteins into smaller parts and it basically acts on the aromatic amino acids. Then we have the carboxypeptidase, which is secreted again by the pancreas as procarboxypeptidase. It removes the terminal amino acids from the peptides, producing free amino acids. Let's see the next two marker. Draw and label the oxygen hemoglobin dissociation curve. This is important. It was asked previously in December 16 and November 2020. So this curve, it explains the affinity of hemoglobin for oxygen. Okay, under normal conditions, the oxygen hemoglobin dissociation curve is S-shaped, yeah, sigmoid shaped. Now this is the percentage of hemoglobin saturation on the vertical axis, and on the horizontal axis you have the partial pressure of oxygen in mm of Hg. So as we know, as the P50 of oxygen is saturation of about hemoglobin to 50%. Okay. This flat curve that you see, this is above 70. This is 70, say 100 mm of Hg. Okay. This is seen in the pulmonary capillaries. The steep part here that you see is seen in the systemic capillaries. In the pulmonary capillaries, where there is minimum reduction in the oxygen transport until the partial pressure falls below 50. So there is not much change, even if there is increased or decreased pressure. While in the systemic capillaries, a small drop will result in a release of large amount of oxygen. This part. Usually the curve is flat above 60 mm. So now, whenever there is any change in the partial pressure of oxygen of about 50%, there is a shift to the left or to the right. The shift to the left is usually seen, okay, when there is lower P50, partial pressure of oxygen is low. Okay, so there is increased affinity of hemoglobin to oxygen. This is seen when there is decrease in temperature, increase in the oxygen, and also there is increase in the pH. The shift to the right, okay, is seen when there is decreased affinity of oxygen to hemoglobin. This occurs when the pH is low, the temperature is high, and there is increased concentration of carbon monoxide or dioxide. Two marker that we have is define Chronaxie and Rheobase. Rheobase we will see first because it is related to Chronaxie. It is defined as the minimal intensity of an electric current that is applied indefinitely, for an infinite duration, that will excite a nerve or a muscle to produce a response. The chronaxia is that it's the minimum duration of a stimulus or the pulse width that is required to excite a tissue when a current strength is set to twice the value of the Rheobase. So this completes your 2025 October physiology paper. Stay tuned for our 2025 human anatomy paper. Like, share, and subscribe our channel for more such PYQ discussion videos. You can also join our Telegram channel to find question papers, solved PDFs, and full video description links.